like to start by thanking the organizers for inviting me to be here and talk about some of the data that we have on the lab. I also like to start by the acknowledgement. I think it is, sometimes we forget that at the end. I want to talk about the fact that the work that we've done requires a lot of people, so there's a lot of collaborators there that have been instrumental in getting the data that I'm going to talk about today. I also like to thank acknowledge my lab members and the funding agents. The work that I'm presenting is funded by NIAAA and CFAS. And finally, I'd like to thank all the subjects that donate their time and biological samples so we could do the research that we're doing. And finally, I just want to say that I don't have any conflict of interest. We heard this morning from the ALC, we heard from Claire that individuals with FASD, they have higher vulnerability for mental health problems. So this I'm showing here is data from Seattle, the original cohort of individuals that Anne Strife was collected, and her data indicated that about 95% of those individuals will show some type of mental health at some point in their lives. This is extremely high when you compare to this same overall data in the general population that is about 30%. But another thing that came with the ALC is, and also Claire talked about just now, is that it's not just mental health problems. There are a lot of physical health problems that is high. So in this chapter published by Emily, Miles, and CJ, it shows that there's a lot of health problems. I'm just showing here a couple of them, for example. So my research is trying to understand a possible mechanism by which alcohol is inducing this high vulnerability to health problems. And we think that the immune system has something to do with it. We always think about the immune system as that system that is there to defend us. When we talk a lot about it during COVID. COVID-19, we talk about increasing cytokines, that this like the chemicals that mediate the mediators in the immune system. But the immune system does much more than just the defense, what is quite important. The immune system is also important for the brain development. It plays a very critical role in brain development and brain function in adulthood. So here there are just four functions in which the immune system is actively mediating or affecting in a typical brain function in adulthood. There is also what is called the fetal cytokine imbalance hypothesis that indicates that for a typical organism development, it is required that there is a balance between the pro-inflammatory and the anti-inflammatory side of the immune system. So if you have any imbalance for the anti-inflammatory or for the pro-inflammatory, you're going to have a altered development in the brain, in the body, that will eventually lead to some type of behavioral change. And what we think that's happening is that alcohol exposure, it is inducing this imbalance to the pro-inflammatory side, and that is mediating some of those alterations in development. And we start this trying to answer that question: does prenatal alcohol exposure affect brain development or the development of the immune system, using an animal model? So this is the work of Tamara Bodner that was done in Joanne Warrenberg lab. And what we did there is that we exposed pregnant rats to alcohol throughout the entire pregnancy, and the control rats just get pellet version of that diet, and we look at the offspring. And we look at the offspring, and one of the things that we look at is just basic markers of the immune system. So I'm showing here two of them, that is TNF-alpha and interferon gamma. Those are pro-inflammatory cytokines. And you can see in the black bars, that is those animals are exposed to alcohol, they have a higher levels of those pro-inflammatory markers. This is in the blood. But you also can see a similar effect in the brain. The same markers, and here I'm showing hippocampus, that is a brain involving cognitive function, emotional function. You see that there is increase in those markers in the brain. Okay, and this work, by the way, was done in Joan Warnberg lab, but there's other labs that have shown a similar profile in animal models. But the next question was, does this actually happen also in humans? So we start collaborating with Dr. Tina Chambers from UCSD, and she has a cohort of a longitudinal cohort in Ukraine, in which she sample or recruit pregnant women that drink alcohol or not, and she follows the pregnancy and the kids, and I think the kids are around teenager now. And we were able to obtain samples of those kids about 2 to 3.5 years old, and we measure about those cytokines. In this case here, we measure 40 of those cytokines. One thing that is important that was done in that project is that they also have neuroassessment of those kids using the Baileys. So we could subdivide our groups of individuals that are exposed to alcohol and the controls in the ones that are having typical development and the ones that are having some type of neurodevelopmental delay. And this is what I'm showing here. This is a complex figure. We're going to break it down. There are 4 groups there. There's the control individuals that were exposed to alcohol. There's the control individuals that are not exposed to alcohol. They're having typical development. There are the control individuals that are not exposed to alcohol. They're having neurodevelopmental delay. And the same thing for the ones that are exposed to alcohol. And we use a technique that allows us to look at networks of cytokines. And there are 3 different networks there. And the first one on top, and the colors blue here means when it's darker means it's activated. So those immune markers are activated. The first immune network we're seeing highlighted in red there, it's activating in all those kids that are never exposed to alcohol, but they're having neurodevelopmental delays. Those markers are also, other researchers have shown that those markers are increased in individuals that have other types of neurodevelopmental disorders. But for us, the most important networks are the 2 following networks. So the third network, there are 3 cytokines that are activated in all the kids that are exposed to alcohol, regardless of their neurodevelopmental status. So that is kind of could possibly be used as a possible biomarker of alcohol exposure. Then the second network is very important because that network we're able to differentiate those kids that were exposed to alcohol and are going to have neurodevelopmental delay from those that were exposed to alcohol but having typical development. So hopefully with this, we can indicate that exposure to alcohol during gestation is affecting the development and the function of the immune system in those kids. But the next question was, is this long-lasting? If we think that those alterations in the immune system is mediating some of those health problems, those alterations have to last until adulthood. So then the cohort that Claire just described, we collect, Claire described the cohorts of Atlanta and Seattle. We also have a cohort that was being recruited in Vancouver, Canada. And there are adults older than 22. Claire described a lot of the outcomes that we assessed on them. And again, we took blood samples to look at their immune function. And in this case, we measure the same markers that we look at in the kids, but now here in the adults. I'm showing the data in a different format here. The data here is in a heat map, and what you can see is in each row is a different immune marker, and each column is a different group. The first 2 are controls from Canada, and the second column is individuals exposed to alcohol from Canada. And you can see that there are different patches in which there are darker blues, indicate that they're increasing cytokines, and there are patches in which they're lighter blues, means that the exposure to alcohol induce a reduction of some cytokines. The next 2 columns are the ones from Seattle, And you see that similar to Vancouver, Canada, there are some patches that are darker blue and some patches that are lighter blue, indicating again that exposure to alcohol is altering the immune system in those individuals in Seattle. And finally, Atlanta, we see a similar variation in darker blues and lighter blues, indicating that the immune system function of those individuals are altered. But what we can see as well is that this preliminary analysis here is showing that those alterations are kind of specific to their site. So there are something there that are making those immune profiles a little bit different. The overall goal is to figure out if there is a unique marker, so we need to increase our screening to figure out, increase our power to see is there a marker that will be able to differentiate those who are exposed to alcohol from those that not across all kind of sites. But again, this is a heat map. It is just a visual representation of the data. So we went on to actually do analysis of each individual cytokine. And what we saw is that from the individuals from Seattle here, we have increased levels of several cytokines in those who are exposed to alcohol compared to controls. The first one on the top is CRP, that is a C-reactive protein, that is a marker of chronic inflammation. Then there are other markers such as TNF-alpha, CCL26, CCL22, IL-17. Those are all pro-inflammatory markers. We are not necessarily seeing the same inflammation in those individuals of Atlanta, but at least those individuals here in Seattle that was part of the study here, they are seeing this higher inflammation in adulthood. But the goal here is to see, are those inflammations that we're seeing here mediating some of those health problems that Claire described? So we see that there's this low inflammatory, lifelong inflammatory bias that's happening. And we believe that this is actually increasing their, mediating the increasing vulnerability that those individuals have for physical health problems and mental health problems. So one of the things that we did to assess physical, besides the questionnaire that Claire talked to you about, one of the things that we did is that we have established a preclinical questionnaire to evaluate autoimmune disorders. So we didn't think that the age in which our individuals, the age of the individuals recruiting, they were actually going to have a lot of autoimmune disorders. So we developed a preclinical screening to see are they going on to develop autoimmune disorders. And what you can see here is that in Atlanta that is the case. Those individuals here from Atlanta, they have higher scores in that screening, indicating that if they already don't have autoimmune disorders, they might go on to develop some autoimmune disorder. But we don't see that, again, in Atlanta. When we try to connect this with our immune markers, we see that IL-15, that is a pro-inflammatory cytokine that is involved in the pathophysiology of autoimmune disorders. We see that this IL-15 is predicting those autoimmune scores here in the individuals from Seattle. We also look at mental health. And in Seattle, we see, again, higher levels of anxiety and depression. And what we also see is that IL-17 and CCL22, again, two immune markers that are pro-inflammatory, they are predictive of those mental health problems that we're seeing here in Seattle. And in the clinical literature, just evaluating depression and anxiety, we see that those markers are actually predictive or involved in the pathophysiology of those disorders. So just a summary to what I told you today is that our data show from the animal model to our clinical cohorts that prenatal alcohol exposure is inducing this long-lasting change in the immune system that we think that might be mediating some of the health problems that we're seeing in this population. To answer the question that was for Claire, We are actually recruiting individuals. If you are from the Calgary region and or if you are from there and you're exposed to alcohol, or if you were not exposed to alcohol and you want to participate, if you know someone from the Calgary region that you think that might be a good subject for us, please, you can scan the QR code. If you're not able, you can just come talk to me and I will provide you information about the study that we're conducting. Final slide is that a lot of this work is done by or supported by the CFAS, and CFAS has several iterations, and there are a lot of data that if you want to access to try to answer some of the questions that you have, here's the QR code that you can access and see what is the process that you need to do to be able to assess the data. Again, if you're not able to read the QR code, you can there are a lot of CFAS researchers here. They're going to be very happy to let you know how you can access this data. With that, I thank you for listening, and do we have time for questions?
Yep. Thank you, Charlie. Yeah, so we do have a few minutes for questions.
My name is Moheb, and I'm a key worker working at Fraser Valley. I've been working with the kids who have fetal alcohol for over 7 years. I saw hundreds of them, and I do also researches about FSD. So I found also recently a research was done by NITO talking about paternal exposure to alcohol before pregnancy can cause symptoms like FSD, and also maternal egg can be impacted by alcohol before pregnancy. Mm-hmm. So when we gonna change our diagnosis based not just for the mom during pregnancy, but going before pregnancy for parents with their dad or mom, because the baby doesn't come from mom only, comes from dad and mom, which is the 23 chromosomes. And also another question, when both parents, they already diagnosed, we suggested that scenario, if the both parents diagnosed with FSD, the child will come with the same disability or will be different? Thank you.
So I think there are some preclinical studies looking at how paternal exposure to alcohol is affecting the offspring, and how there's also preclinical studies from a group in Australia that look at around pregnancy, how those alcohol consumption might affect pregnancy. And there are some effects that come from paternal exposure. But for me, I think one thing that's important is that I think a role that the father can play as well is that if the woman's pregnant and the father continues to drink and is not supportive as it should, so I think that is another important role that people in the social context could play as well.
Maybe one more question, Chris.
It's just beautiful, and having looked at the data, knowing being responsible for recruiting for the Vancouver site, I'd like to reflect on what happened with our NeuroDevNet study, and I'm just wondering if this difference of Seattle, because it's interesting that Vancouver and Atlanta are showing similar sort of cytokine the blue patches are reversed in the same order, but Seattle, and I'd like to think of perhaps, I know that we sent kids to Seattle to be part of that study that were more stable families, and that there may have been differences. Claire called this a long time ago, but the difference between being reared with your family versus being taken out of your culture, taken out, so that as we look at this data more and more to actually see what the social determinants or social drivers might have been that might have impacted these individuals prenatally as well as in their rearing environment postnatally. That's my thought. I'm having trouble with Claire, and I didn't know how to answer the question. It's just not clinically fit with me that ACEs are driving depression. I also think I've just seen kids who've been raised without ACEs, but seem to be higher risk, and I wondered if that was that either genetic family history, that there's just something, I know the data shows it, but it just doesn't fit. That I still believe alcohol has something to do with depression. So.
Yeah, that's not on. I think that we believe that.
Could we get the...
Please, at the microphone for Claire.
Is it on now? Yeah.
Yeah.
I think, and I believe very strongly that anxiety in particular is associated with FASD. Data. So we're going. It wasn't that they didn't have. There was indirect effect of the alcohol, and that's in a published paper. We did a path analysis and found that, but it could be that the ACEs overwhelmed the other.
Actually, one more question? Yeah.
I know that we all hope that all of the research and data collection will eventually lead to, services for folks, but I guess a more specific question is, do you feel like there's a potential for this data to be connected directly to pre-service in the way that physicians are trained and educated? Because when we're talking about the reality that it's a whole body disease and so many folks are going to be seen disproportionately by specialists, I wonder if you've had opportunity to present, for any of you really, this data in relationship to medical schooling and the way that physicians in specialties where you see disproportionate rates of folks that have FASD, so that they can be a part of the overall potential identification for folks that might not know about their alcohol exposure or their FASD diagnosis.
Yeah, I think so. I think I think there's a missed opportunity in med schools to inform the future doctors about what are the possible health problems that are associated with FASD. And I think one possible problem might be that until not long ago, there is not actually many studies in adults with FASD. So there was not this wealth of data that we could knock on the door of the doctors and talk about it. I think the work that has been done by the LC and the work for Claire and many others here, that is like describing this long-lasting effects and seeing how this prenatal exposure is actually having a full body deficits and problems might increase our ability to knock on the door of the med schools and try to raise more of the awareness.
All right. Thank you, Charlize, again.